Okay, so we've looked at eukaryotic cells, which are larger cells make up multicellular animals and plants and higher plants. Let's have a look at prokaryotes. Sorry now. So here is a prokaryote, which is bacteria. Okay, listed bacteria and algae are prokaryotes. Let's think about E. coli, which is Escherichia coli. It's called really. It's a bacteria which is found in very large quantities in your gut. It's most strains of E. coli are completely harmless, and in fact, they're kind of good for you because you need a flora of bacteria in your gut to stop other nasty bacteria getting in there. There are a few strains of them which cause food poisoning and so on. Mostly they're harmless, if not beneficial. Right, and here's a red blood cell, just to give you a bit of a comparison in size. Okay, so red blood cells about 6 microns across. That's 6 times 10 to the minus 6 meters. For E. coli, about 1 or 2 microns long and 1/4 of a micron wide, so a lot smaller. And we've also seen that probably mitochondria in eukaryotes were probably originally prokaryotic cells in some kind of symbiotic relationship. So anyway, here is a bit more detail in the shape of a prokaryote of an E. coli. Right, this blue thing here is the chromosome, and it's made of DNA. It's circular, unlike eukaryotic chromosomes, which are linear. There's no protein associated with that. It's kind of naked DNA, which is unlike eukaryotes, which have proteins called histones associated with them. It's actually reasonably big. I think it is 3.6 times 10 to the 6, 3.6 million base pairs long. Okay. And the size of the human genome is apparently 4 times 10 to the 9. So it's about 1,000th of the size of the human genome. Most of that E. coli genome actually codes for proteins, which is not the case for humans, where it's like only 98% of it is non-coding. So there's a much lower level of organization of the DNA in prokaryotes. Okay, so what else? Right, you also have these little bits of DNA, and they're called plasmids. Okay, and they are transferable, right? The cells get a bit leaky from time to time, and the plasmid may go out of one cell to another bacteria, and that unfortunately is a way in which antibiotic resistance is spread very quickly through a population of microbes. If the resistance gene is on a plasmid, and then the plasmid can spread to other ones. Let's just think about sexual reproduction. Prokaryotes, do they have sex? Well, they do actually, not very often. Most of the time they just divide asexually, and they can do that very quickly, once every 20 minutes under ideal conditions. But sometimes they can also, they form like little gaps in the cells, and they join up with another one, and they exchange a little bit of chromosome. So they swap bits of the chromosome DNA from the main chromosome. So they do sexual reproduction, so they can transfer genes between individuals, and they can also transfer genes, of course, with the plasmids. This thing here looks like a tail, is called a flagellum. Not all bacteria have them. Some do, and it's used for basically moving the bacteria about. I think, have you heard of a bacteria called Helicobacter, which causes stomach ulcers in human beings? And it uses that flagellum. It infects the cells, the epithelial cells lining the stomach, and your stomach have a very thick mucus layer, and it uses that flagellum to force its way through the mucus layer and infect the cells. So that's used for moving. The other thing I haven't mentioned is the cell wall. It's not made of cellulose. It's made of a substance called murine, which is a glycan. You may see the word glycoprotein, which is not quite the same as that. A glycoprotein is a protein with a little bit of carbohydrate on the end of it, whereas this thing is really, it's mostly carbohydrate, the glycan bit, with a few very short peptides, 2 or 3 amino acids long stuck to it. It forms a kind of a mesh structure, and it stops, very much like the cellular cell wall, it will stop bacteria bursting through osmotic pressure. So if you put them in water, like red blood cell, if you put that in water, the osmosis, the water will enter the cell and the cell will burst. Bacteria won't, a plant cell won't, because it's got this cell wall here. Okay. The disruption of manufacture of the cell wall. So if you stop the cell wall, you can kill the bacteria. And penicillin, which is of course an antibiotic, that's how penicillin works. It disrupts the formation of the peptidoglycan. It forms a cell wall, and this is the E. coli, or the bacteria, whatever kind of bacteria it is, can be killed. Okay, so that's enough of all we need to know of prokaryotic cells. What we do need to know about though is viruses. Okay, now there's a few pictures here, so just ignore some of them for the time being. All right. Okay, so right, this, we just look at this picture here first of all, and ignore all the snooker balls on the left. Right, this is a viral particle. Okay, now viruses, whether they're living or not, is debatable, because they can't reproduce themselves. They have to hijack living cells, hijack the machinery of that living cell in order to make more viruses. So they don't respire, obviously, and they don't excrete and all those sort of things. But well, let's see what they can do. Right. So that black thing in the middle there is the viral genome. Now, in the case of HIV, that is, let's stick with HIV. So in the case of HIV, this is RNA. It's not DNA, it's single-stranded RNA. And the coronavirus, which causes SARS-CoV-2, that virus, that's also single-stranded RNA. It's not a DNA, but some deep genome. Some viruses do have DNA genomes. And in viruses, the genome is always coated with these red balls, which this is the capsid. Okay, now the capsid is made up of one protein, and they fit together very tightly to form this protective layer around the genome. And this is what I'm trying to draw here. Now, the capsid can be of 2 types. It can be like this one here, which is an icosahedron. So what happens is these little red balls, so this is one, they form equilateral triangles, okay, like that one there. Then that equilateral triangle, that goes in there, and then you build up the icosahedron from all of those little equilateral triangles, and it forms, an icosahedron is a 20-sided figure. It's roughly spherical in shape, and the viral DNA or the RNA sits inside there and is protected. There may be a few proteins in there as well. The virus needs to get going straight away, like a RNA polymerase, which causes the RNA to be copied. Right. The other kind of viral capsid is this one here, which is called the helical. So there, again, you've only got one kind of protein subunit, the little red ball, and they arrange to form a spiral shape, and you have your RNA or your DNA sits in there. The coronavirus has this kind of capsid, whereas HIV, the human immunodeficiency virus, the one causes AIDS, that has an icosahedral capsid. Okay. Right, now in some viruses, that's all you have. So you just have the RNA or the DNA in the capsid. But most viruses which affect animals have got this blue layer around them here, that's a cell membrane. It's a phospholipid bilayer, and it's been stolen from the host cell. So the virus doesn't make that; it just hijacks a bit of the host cell's plasma cell membrane, and in order to protect itself. So we just very quickly have a look what will the virus do. So here's what we've got: a eukaryotic cell. The virus comes along. I just draw it as a red. I draw it as a blue blob, so I don't get confused with the capsid protein. So that somehow it sticks to the surface of the membrane. We'll talk about that in a minute. It gets in. It's got a genome. It's got its own genes, and it switches off the cell's genes, and it makes the cell's machinery. So the cell's ribosome, protein synthesis, and everything. It turns the cell's machinery over to making lots and lots of copies of itself. Once the cell is full up with copies of the virus, the cell dies, bursts open, all of these viruses come out, and they can go and infect other cells. Okay, so the virus, as I say, is not strictly speaking living in itself. It needs a cell to go and live in. Right, let's get back to over here then. Now, let's talk about how viruses actually get into cells here. Okay, so it's very dependent on this. See this green protein? This is a transmembrane protein. So it's in the phospholipid bilayer, and this is sometimes called the spike protein, which is quite infamous in the coronavirus. The case of the coronavirus, and that's why they're called coronaviruses, because you can see these little spikes in the electron microscope picture. It looks like a crown. Right, these spike proteins recognize something on the surface of the cell, on the surface of the host cell. So here is our virus now. Now, if it is a coronavirus, the red thing it recognizes on the host cell, is the host, is called the angiotensin converting enzyme, sometimes called ACE. And if you have heard of people who've got high blood pressure, they sometimes take ACE inhibitors. So these are drugs which inhibit that, and that lowers their blood pressure. So it's nothing to do with the virus. The virus has just hijacked it. It can see it's something that it can bind to, and that's what it does. Right. In the case of HIV, it doesn't bind to to the ACE. It binds to CD4, which is a protein found on the surface of T helper cells. And it's also on the surface of, in less quantities, on some macrophages as well. So CD4 is on the surface of T helper cells, so HIV infects the T lymphocytes, and that's why it damages the immune system. The coronavirus, it binds to ACE, and ACE is on the cells, the epithelial cells of the lungs in very large quantities, so it can get into your lungs very easily, get into your body that way. And then most cells in the body also have ACE on the surface of them, so it can infect all of those. That's why people who get very ill with coronavirus very often get all kinds of things going wrong with them, things going wrong with their kidneys and everything, because a lot of cells have ACE proteins on the surface, so the virus can infect them. So, right, the virus is bound to, using the spike protein, it's bound to the receptor on the host cell. And then one of two things can happen here. Okay, now, one thing can happen is, so draw an arrow there, is this can cause fusion of the blue membrane of the virus and the black membrane of the host cell. And see, so the membrane is fused, and as a result of that, our little package of DNA or RNA has got into the cell where it can, and that's the viral genome, and it can do its thing. Still surrounded by the little red bits with its capsid proteins. It might have a few other little proteins in there it needs to have to get started. Right, so that's the way HIV gets in. It causes membrane fusion, okay, and it gets straight in that way. Coronaviruses, just as an example, they're a little bit different. What they do is they can't the blue membrane can't fuse with the host membrane, and what happens is it's in there. It's been phagocytosed, and the host cell will, of course, you get little lysosomes will fuse with that, and they contain like proteolytic enzymes. Unfortunately, those proteolytic enzymes don't actually harm the virus; they just help it get going. And then, so anyway, the RNA has still got into the cell, or isn't the RNA in the case of a coronavirus? It could be DNA. If it's a DNA virus, has got inside the host cell, and there it can go and do its damage. One last thing is before let's do this little comparison here. Let's see what happens here of the two viruses. I've just chosen these two viruses. The HIV you do need to know about. The SARS-CoV-2 chosen obviously because it's interesting. Genome. HIV is single-stranded RNA. Same with the coronavirus, single-stranded RNA. The capsid, in the case of the HIV, the capsid proteins form that icosahedron. And in this one, they form a helix. So they're a little bit different there. The spike protein on the outside of HIV, that binds to the CD4 receptor on the T helper cells. In SARS-CoV, it binds to the ACE, angiotensin converting enzyme. It's just purely opportunistic. It's nothing to do with that enzyme. The cells infected are lung epithelial cells and a lot of other cells also. Entry into the cell, right, the HIV, it does it by membrane fusion. Whereas the coronavirus, that gets in by phagocytosis. The life cycle, all right, what do I mean by that? Well, with SARS-CoV-2, the RNA gets copied, and the RNA gets transcribed. That means you make proteins from it. And then the proteins and the RNA which have been made are packaged in the capsid, and then they steal a bit of the cell membrane, and then you've got your virus particles. The HIV life cycle is different. It's, as you know, that HIV can take years and years to develop after infection, whereas coronavirus is pretty quick. So the life cycle of the HIV, the RNA, you make a DNA copy of that. They make it using an enzyme called reverse transcriptase. Makes a DNA copy for the base pairs, and the DNA is then incorporated into the host DNA. And there it can sit not doing anything for a number of years, or maybe not as long as that. And then something happens, and all of a sudden that DNA will be transcribed. You'll make HIV, it will be transcribed. So you'll make HIV proteins instead of the normal proteins for the cell, and then the virus can be assembled, and it can go and cause more infections in other cells. Okay. Right, so that is viruses for you.